In 1969, inside the National Institutes of Health, physicians Joseph Fraumeni and Frederick Li noticed a terrifying pattern. They were looking at 4 distinct families where cancer was striking with relentless frequency. At the time, the medical establishment viewed cancer as a disease of aging or a random stroke of bad luck. The idea that a massive, multi-cancer susceptibility could be directly inherited from your parents contradicted the core understanding of how tumors formed. But the patients in Fraumeni and Li's files didn't fit the random model. They were seeing very young children developing rare soft tissue sarcomas, while their parents were diagnosed with breast or brain cancer in their 20s and 30s. This is the pedigree chart from their defining publication. The density of black markers maps a hereditary tragedy, Li-Fraumeni Syndrome. Looking closer, half-filled circles mark soft tissue sarcomas, split-filled circles mark breast cancer, and crosses indicate family members who died across multiple generations. It took decades for molecular biology to catch up. This DNA chromatogram shows exactly what went wrong. The top sequence is the healthy control. On the bottom, a single substituted letter creates a premature stop codon, breaking a crucial gene known as TP53. TP53 normally acts as the guardian at the genome, repairing DNA damage or forcing a damaged cell to self-destruct. Fraumeni and Li proved that when a person is born with this gene already broken, severe cancer susceptibility is hard-coded into their DNA. Today, a diagnosis of Li-Fraumeni syndrome carries an almost 100% lifetime risk of developing cancer. The threat is constant, and the medical response must be equally aggressive. To manage this risk, patients undergo the Toronto Protocol. This is a grueling, lifelong regimen of whole-body MRIs, targeted ultrasounds, and frequent blood tests designed to catch tumors at their earliest, most treatable stages. The surveillance demands that very young children undergo general anesthesia for their annual scans, inflicting chronic anxiety and an immense psychological toll on entire families as they wait for the next test result. Researchers are developing new technological workarounds. Liquid biopsies offer a way to detect microscopic DNA fragments left behind by cancerous cells in a standard blood draw. This can identify a developing cancer months before a tumor becomes visible on an MRI. Parallel research focuses on chemoprevention. Clinical trials are testing drugs like metformin, a common diabetes medication, to see if normalizing metabolic abnormalities can delay or even prevent tumor onset in TP53 mutation carriers. Identifying the TP53 mutation answered the question for a handful of heavily affected families. But it also raised a much larger question. If these patients get cancer because they inherited a broken guardian gene, what is breaking that same gene in the rest of the population? Joseph Fraumeni scaled his focus outward. He wanted to know what external forces could fracture the DNA of people born with completely healthy genomes. This is the core of environmental carcinogenesis. It studies how outside agents, like chemicals, radiation, or viruses, inflict DNA damage faster than a cell's natural repair mechanisms can fix it. Instead of tracing individual family trees, Fraumeni began mapping massive populations. By plotting cancer mortality across the United States, his team could identify geographic hotspots where disease rates spiked alongside local industrial activity or water contamination. They isolated workplace exposures through occupational cohort studies. By comparing the specific cancer rates of shipyard workers or plastic manufacturers against the general public, epidemiologists could pinpoint exactly which environments were toxic. These studies uncovered undeniable links between specific traits and fatal illnesses. The data proved that asbestos exposure in manufacturing plants and construction sites was directly driving a lethal spike in lung cancers and mesothelioma among workers. To separate genetic fate from environmental risk, researchers utilized migrant studies. When a population moves between countries with different baseline cancer rates and their own disease risk shifts to match their new home, it isolates the environment as the culprit. These mechanisms aren't limited to industrial hazards. Broad lifestyle choices like diet and smoking operate as widespread, self-inflicted environmental exposures that slowly overwhelm cellular repair over decades. Inherited genetic mutations are locked in at birth. Environmental exposures are modifiable. By proving exactly which chemicals and environments cause DNA damage, environmental carcinogenesis created a direct path to public health regulations that save millions of lives. For decades, cancer research operated on two parallel tracks. Geneticists studied rare inherited traits, while epidemiologists studied population-level environmental hazards. Genomic epidemiology merges these two fields. It utilizes massive computing power to combine the health records, environmental exposure data, and full genomes of thousands of people into a single analytical framework. Classical genetics looks for one rare, high-penetrance mutation that guarantees disease. Genomic epidemiology scans for thousands of common, low-risk genetic variations spread across a population. Each tiny variant contributes a small amount to an individual's overall polygenic risk score. This reveals the mechanics of gene-environment interaction. These subtle genetic variations dictate exactly how vulnerable a specific person's cells are to specific environmental toxins. If two individuals face the exact same exposure, like identical years working in a chemical plant, one might possess a metabolic gene variant that makes them highly susceptible to the carcinogen, while the other's genetic profile successfully clears the toxin from their system. Joseph Fraumeni's career traces this exact evolution. He started with the microscopic tragedy of a single family's broken DNA, expanded to map the environmental hazards of industrial towns, and finished by building international consortia to study polygenic risk across global populations. By integrating the microscopic and the environmental, the architecture of susceptibility is finally visible. We can move past treating cancer as an inevitable reaction and build proactive, personalized strategies to stop the damage before it starts.